All right. Good afternoon, everyone. Thanks so much for joining us. We're really glad to have you here and really excited about today's discussion. My name's Tim McPeak, and I will be moderating the discussion today. And we're excited to have a conversation today about something that really doesn't always get talked about openly in clinical research, which is the fact that trial outcomes are rarely determined by the target biology all by itself. Outcomes are also a function of the clinical trial design as well as the operational execution of the trial itself. Even with advances in technology and analytics, many of the risks that impact studies still come from everyday operational realities at clinical sites, things that are often difficult to see until late in a program. In this conversation, we'll talk about why those challenges persist, what they look like in real world drug development, and how sponsors are starting to think differently about oversight and visibility during trial execution. Before we get going, just a couple of quick housekeeping notes. We would love to hear your thoughts and questions as we go. If you do have any to share with us, please drop them into the chat down at the bottom of the screen. There is also a Q&A box, but we would prefer just to keep it consistent if you would use the chat function. It should be kind of right in the middle of your screen on the in the interface. We're also going to run a couple of quick polls. They're going to be anonymous polls, so we would really appreciate your participation to get some insight into your experiences and also drive some of the discussion as we go. So please, again, feel free to participate as those appear. We'll tell you when they're coming. And without further ado, I'm really excited to introduce our speaker today, Dr. Rick Graham. Rick is a serial entrepreneur, board member, and former head of research and development for a publicly traded company, bringing 3 decades of experience in drug development and R&D leadership. Over the course of his career, he's contributed to development and approval of 5 marketed therapies and led programs through late-stage clinical development across major therapeutic areas. His work has consistently focused on strategic alignment of high-quality science with rigorous trial design and operational execution, experience that ultimately led him to found TruTechnologies. Rick, it's always great to see you. Thanks so much for being here today.
Yeah, thanks, Tim. Thanks for spending time, and thanks for making me sound really old with that introduction.
Absolutely. Yes, my pleasure. So look, I think as we get going, as often is the case, starting at the beginning might be a good place to kick things off here. So can you tell us a little bit about, but and again, prior to co-founding TruTech, which we're certainly going to get to, tell us a little bit about your history, about your career trajectory, and how you ended up where you are today.
Yeah, sure, sure. Grew up in the Midwest, in a small town in eastern Iowa. Ended up going to college at Iowa State University, where I studied biochemistry and biophysics. Worked for a few years at a small CRO in Kansas City, which really sort of got me the drug development bug. That's where I learned drug metabolism, and then decided to pursue my PhD in clinical pharmacology at the #1 pharmacy school in the nation, UNC Chapel Hill.
I'd
Um,
like to say go Heels, Rick, but I'm not really a fan, so I'm going to leave that
Yeah,
to you here.
yeah. Thanks. We've got a little rivalry here. Once I finished with my graduate education, I worked for GlaxoSmithKline for a period of time in Research Triangle Park, North Carolina. I then moved to the West Coast and took a job with Genentech, which was then acquired by Roche. So I was there for 7 years. Worked at a company called Onyx Pharmaceuticals, which was acquired by Amgen. I was head of research and development at Theravance Biopharma and had co-founded another company in the meantime. In addition to TruLab, which was founded 6 years ago, now TruTechnologies, which is a clinical trial technology company.
That's great. Yeah, and we'll certainly dig into a little bit more about kind of the why and how it happened behind co-founding TruTechnologies. But would love to talk a little bit about, kind of you did a great job kind of outlining your career and history. What would you say has changed in drug development over the years? And then I'm probably going to follow up with that to maybe talk a little bit about what has stayed the same.
Yeah, yeah. Yeah, now you're going to test my memory, but I would
Yeah.
say that, one of the biggest changes really has been the emergence of new therapeutic modalities over the years. When I started a long time ago, the focus was really small molecules, so chemicals that are turned into therapeutics. Then it sort of switched over to excitement around monoclonal antibodies. And since then, antibody drug conjugates, which is the mixture of an antibody and a small molecule, and now multi-specific antibodies, where you're hitting multiple targets at the same time instead of a monoclonal, which just hits one, and then all kinds of other complex biologics. So quite an evolution over the years in terms of the quality of science that I've seen change.
And then just following up on that, what do you feel like has kind of not changed or has stayed largely the same over the same time period?
Yeah, I mean, it's pretty interesting because the science, like I said, is really advanced significantly. I can't even think about how many blockbuster drugs have been approved during the course of my career, which is amazing and helps patients. But the way the trials are conducted, especially at clinical trial sites around the world, in many ways has stayed the same. And this is really what's in at least in my mind, has been something that we've been very, very slow and sometimes reluctant to take action on. And what that means is there are major inefficiencies in how we conduct clinical trial research and therefore opportunities for improvement.
And so, if you had to speculate, like why things haven't really changed, like what are some of the forces that come to mind in thinking about why, with so much, especially on the science side, with so much incredible breakthroughs happening, why do you think some of these things have been reluctant or slow to change over time?
I mean, it's hard to say sort of on the population level why, but if I sort of just think through why it might be, we tend to control things or change things that are in our control. And oftentimes, discovery and early development of a drug happens at the biotech or pharma company. And then when you get the data from the clinical trial, the analysis happens at a biotech or pharma company. All the stuff in the middle, honestly, I think it's just an everyone's problem and no one's problem situation. People are like, well, somebody else must be taking care of that. And in my career, I used to think that until I realized that wasn't true, so I did something about it.
Yeah, it's interesting. I'm still relatively new to this space, but one of the things I've been struck by, particularly with my time with TruTechnologies, has been kind of the broader ecosystem around these trials and how many different players there are, and just sort of different stakeholders, and that I think that diffusion of responsibility you're referring to seems like kind of a plausible explanation, right? It's both when it kind of is diffused across everybody, it's not really one party's responsibility to fix it. It also, I think, allows parties to kind of be like, hey, nothing I can really do about it, right? So
Exactly.
interesting. So to kind of keep moving, as you look at the biotechs and as you look at the biotech industry, obviously, there's regularly cycles that we see, but how do you kind of view the state of the biotechs today?
Yeah, I mean, I think it's re really good in terms of how we're starting off 2026, and I'm been to back-to-back conferences the last few weeks, and the sentiment seems very strong. So fingers crossed. But if I generalize it more, I would say that there, there's constantly tremendous pressure on operators to return capital to investors and therefore their shareholders. At least over my career, there's unprecedented competition for the same therapeutic targets across all the different therapeutic areas that we focus on. So companies are competing with each other, and then of course we hear a lot about the competition from China. So there's just this unbelievable emphasis on clinical trial execution speed and taking efforts to improve the probability of technical and regulatory success. And again, it goes back to what I said earlier. Like there are things that companies can do internally, and they are doing them, and they should be doing them, that can help with speed and probability of success. But let's also pay attention to the things that we're not paying attention to, which is what my job is.
Yeah, that makes sense. You mentioned spending some time at some conferences. I have a strong suspicion that perhaps AI was a topic at some of those conferences.
Yeah, how'd you know?
It just seems like it's just in the air right now. I'm not sure. We have to talk about AI in a webinar because I'm not sure you're allowed to do a webinar in 2026 without touching on AI. But while we're on the topic, I'm curious. It sounds like you probably are hearing about it. Are you finding that it's on a lot of people's minds, and was it a topic of conversation with a lot of people you interacted with?
Yeah. Good question. It was. So let me take these in two parts. The one conference I attended was more of an investor sort of partnering conference, and two main topics. One was AI in drug discovery, not in drug development per se, but more in drug discovery. And then the other was around running clinical trials in China. So put that aside for now, but let's
Okay.
talk about your question, which is AI. I mean, it's unbelievable, the great work that people are doing. Really smart people spending a lot of time and a lot of money on identifying new disease targets through AI and computational tools, designing molecules that are beautifully optimized to hit a target that we thought was maybe undruggable in the past. This is especially true for antibodies that I talked about earlier and other protein therapeutics. And this is all before clinical testing begins, right? So tremendous amount of progress, effort, money being spent in early drug discovery to optimize the molecule that's going to ultimately go into the clinic. So that was one theme there. Um,
Would you say, sorry to interrupt, but would you say then like that it's more than just hype? Like there's real like material progress
Absolutely.
being made and it's kind of
Oh
changing
yeah,
the game?
yeah, absolutely. I mean, one of the panelists at this company was, or at this conference, is the CEO of a company who has the first truly AI discovered drug, which is now in the clinic. So tremendous progress being made, and it's
Yeah.
really important work, and a lot of really smart people working on it. So put a pin in that for 1 minute.
Yeah.
Let me then go to the other conference that I attended, which is more of a focused conference for clinical pharmacology modeling simulation. And there was a panel on AI, and the focus of that panel was not drug discovery. It was once the drug passes through the clinic and they get the data, pharmacokinetic data, biomarker data, safety data, efficacy data, and then they're talking about using all kinds of really sophisticated artificial intelligence tools to model and simulate that data. Again, really, really important work. But here's, Tim, what struck me in attending both those conferences. There's a paradox. And then paradoxes, and I don't mean to diminish the importance of this work. Both of those things are
Sure.
really important. But a lot of times people go after the shiny new object, right? And sometimes we're ignoring something that's easy to address right there in front of us because we're so distracted this way and this way. And remember we talked earlier about it being out of people's control. I think you
Yeah.
gravitate toward those things that are in your control. But
Sure. It's
what I'm
human
talking
nature. Yeah.
about, what I'm talking about here with operational execution at clinical trial sites, honestly, we don't really even need to think about AI. We're literally just talking about replacing the current state, which is writing important information on pieces of paper or a paper towel or sometimes a glove, and then later entering that manually into an electronic data capture system. So yes, like to summarize all that, AI is super important. Lots of advances in drug discovery, lots of advances in how we analyze and interpret our results. But let's also not forget about the stuff that's also really important, which is executing the trial.
Yeah, it almost hurts your brain a little bit, right, to think about these incredible advancements and just the power of some of the tools that are now available, and contrasting that with literally data on a paper towel or a glove. It's kind of hard to square those things in 2026 and consider that's kind of how we're still doing things.
Yeah.
Um, this is probably a great time. Why don't we use this as a springboard to jump into a poll here? So we'll open up our first poll, and I'm just going to read these so everybody can hear them, and particularly for the recording. Question is, what do you currently believe is the largest challenge in drug development today? Choices are, and we would ask you to just choose one here. First choice is high failure rates and efficacy. The second choice is development costs and long timelines. The third choice would be major protocol deviations. Fourth choice would be clinical trial logistics and biological sample management. And then last choice would be anything that we didn't list here. If you want to mention it, please go ahead and specify in the chat. We're going to open that up and we'll give everybody just a few moments here and then we'll kind of see what people are thinking out there. We don't get to vote, Rick, just in case you're
Oh,
wondering.
thanks.
We're excluded. We don't want to bias
Can I undo my vote?
them. All right, we'll leave that open just a bit longer here. All right, we have got some results here. Let's take a look. Um, Rick, I'm going to ask you for some thoughts here, but I'm going to run through these first. We don't see anybody choosing other, which is great. The top choice by quite a bit here is development costs and long timelines. So again, that's the single largest challenge people are kind of seeing out there. We would see next here 31% trial logistics and sample management, and then pretty close to a tie and for high failure rates and efficacy. So first and foremost, thanks to all for responding to that poll. Appreciate that. Rick, let me bounce it to you. What are your thoughts on that? How does that land for you relative to your experience and what you're seeing out there today?
I probably would have rank ordered them the same in my mind. I think everybody is spot on. The majority of people are spot on with development costs and long timelines. And I'll bet the second one, clinical trial logistics and sample management, if people were given two choices, I'll bet they would have probably pick those two. Those are the big ones, Tim. And I mean, yeah, I mean, with all this competition too, you just think about not only you're putting in a lot of time and money to develop these drugs, but there's still a lot of risk in this industry, and you might actually fail. So, development costs and long timelines are important. Investors always want things done fast, but also really, really well done. And it's
Yeah,
a
and
show
that's
of
the
timing.
balance, right?
Yeah,
Yeah,
it's this
speed
impossible
versus
triangle,
quality.
right?
Yeah.
Impossible triangle is cost, speed, and time. And we always, operators and investors always want on all three. And usually the rule is you can only have two out of the three.
Right. You only get
Um,
two,
you
right?
know,
Yeah.
and then the other one, clinical trial logistics and sample management. I mean, for me, like that's what this is all about. I've already alluded to it. I mean, my as you talked about at the outset, the focus of my career for the most part had been on drug development. So studied clinical pharmacology, got an advanced degree, worked at all these companies to develop drugs. And then I got to a point, and we'll talk more about it later, but I got to a point where I just couldn't take it anymore because I saw what was happening and I had to come
Yeah.
up with a solution, which I've done.
Yeah. Yeah, and there's clearly like at least some relationship between the logistics and sample management to certainly timelines, right? So obviously some of these choices are even related to each other. That's great. Again, thanks to everybody for the poll response. We appreciate the feedback. I think we have at least one more coming up as we go here. But Rick, as we kind of keep moving, we enjoy a mutual friend who is your co-founder, Scott Ogle, is also our CEO. And there is some well-loved mythology within the company about kind of the founding of the company, and kind of how you guys as longtime friends ended up actually going in to be business partners. Could you talk a little bit about the story behind that, kind of just, how all this started and kind of how we've, ended up where we are today?
Yeah, for sure. I love talking about it. I love Scott Ogle. He's one of my favorite people in the world. Um,
Same on that front. Yes.
and kind of what's interesting about this is that, Scott and I have known each other for, gosh, we met in 2002, so well over 20 years. And it turns out that Scott's wife was friends with my wife in middle school, and they were roommates together at Carolina. So that's how I
Okay.
knew him. But only 6 years ago, we came together to start the company. So let me give you the backstory because I think it's pretty interesting and relevant. You mentioned at the opening, Tim, that I had worked on 5 therapeutics that are now on the market. So unbelievably lucky to have done that, and I'm very proud of that accomplishment. What most people don't know is that in 4 of 5 of those cases, drugs that either had BLAs or NDAs approved, there were significant issues with biological samples that came up during regulatory review. So just think about that for a minute. 4 out of 5 of those marketed drugs that I worked on, like that's not by chance.
That's
That
not a
is
coincidence.
absolutely not by
Yes.
chance.
Yes.
That means that there's an egregious situation that should be addressed. Okay. So, in several of those instances, there were cases like the electronic data that we submitted around biological samples. Again, what I'm talking about are PK samples, biomarker samples, safety labs were submitted to the health authority. Health authority said, time out. Your electronic submission doesn't match what's written in your clinical study report, and we need to reconcile that. So that was for me a long, long time ago when I was much younger, and like the person receiving these data, that was the aha for me back then. At one of these companies, I was actually asked to form and lead a task force to map out from the time a sample is collected at a site to the time it was reported in a filing, what happens to it. Like that was the asymmetry that I had, Tim, where it was, like time in your life where you remember something so vividly and it clicks for you and you're like, hey, I'm probably one of a few people in the world that really understand it. Well, that was
Yes.
it. And what do I mean by that was it? What I mean is imagine a process map, like 12 swim lanes of constituents, not people, but like groups, 40 steps, manual on paper that are required from the time that sample is collected to the time it's reported. That was the aha for me. Okay.
So real quick, Rick, how was this received? Like, did you find that as you kind of came back with this flow that people were shocked, or do you think people innately kind of understood it, or was this kind of a revelation for folks to see what
It
actually
was,
happens?
yeah, it was kind of in between. It was a revelation for me. It was for others, I think it was not surprised. Let's put a Band-Aid on this and get through it and move on.
Yeah.
I never let it go, man. Like for 6 years, I tried to give this idea away because I was doing other things. I tried to write to people at companies and say, "Hey, I've got an idea for this problem. I think you should solve it." And it was always like, "Hey, great idea," and then nothing. And so it wasn't until one guy finally listened to me, and that's Scott. So let me come to that in a
Yeah.
minute if you don't mind. But I want to tell another important story, which is there was another situation where we were running a phase 3 trial, probably around 50 sites around the world, and we're measuring pharmacokinetics, drug in the blood of patients that received a dose of this medication or not, right? And it turned out that we were seeing drugs circulating in the blood of people who had never received a dose. So that was a pretty big deal. That was a concern. It was a quality issue. And we investigated what was going on. That shouldn't happen, right? Well, it
Right.
turns out for this particular program, we were also measuring what's called an active metabolite. So that's a conversion product of the parent drug that gets converted by the body into something else. We're measuring that. Doesn't always happen, but in this case, we were. We were also detecting the active metabolite in the blood of people who never received a dose, which is not possible. The body had to convert the drug. Okay, so clearly what was happening is there was evidence of sample switching going on. Clear evidence of sample switching. Now, I mean, we were basically flying blind because we're getting the data, but we're not knowing in real time what's happening at the site. We're relying on this paper trail and data
Right.
that's entered much later. So this was very, very difficult to try to understand. Now, the drug was ultimately approved because the benefit risk was so obvious. The drug worked really well. The adverse event profile was very manageable and very light. But the consequence was that all of the samples that were collected during that phase 3 trial were discarded from the application and ultimately from the drug label. I mean, think about the ethics of that, first of all. Think about the
Yeah.
disservice to physicians and patients that are trying to prescribe the right dose of this drug. And that's when I realized, Tim, that I couldn't take it anymore and I had to do something. And that's when I partnered with Scott. Now, Scott is, we talked a little bit about him, but why is it important that he's the guy that I partnered with? It's because he spent a large part of his career prior to starting TruTechnologies in displacing paper from another adjacent industry, which is the credit lending agency. So Scott is fantastic at taking complicated manual processes that are on paper and turning them into a technology solution that make it easier for people.
I would imagine, Rick, as you started discussing this with Scott, that he might have asked the question, which I think a lot of folks ask, which is like, hey, it's, when you guys were talking, what it was probably 2021, 2022, maybe something like that. Like, why is this problem? Why are we still using paper in 2020, in the early 2020s, so to speak? Like, and if you didn't, it's a question I've certainly asked. So I'm just curious, like, does it go back to that diffusion of responsibility we talked about, where it's kind of not one entity's or person's problem to solve?
Yeah, I think so. I think that's probably it. But what's funny about it is actually when I first brought this issue up to Scott, remember we started the company after a conversation in 2018. We started the company in 2019. But
2019, yeah.
in
Yeah.
that conversation with him, he's like, wait a minute, you guys are supposed to be curing disease. Like we, the public, trust you to be doing things like at a really high standard. I actually don't believe you that this is how things are done. So he actually didn't believe me at first.
Well, it's funny you mention that because early in my tenure here with TruTech, I had the opportunity to go to a clinical site, and I had heard a lot about the problems, and we're using paper, and I won't say I was skeptical, Rick, but I was also like, come on, like it kind of didn't like it didn't really make sense to me. And lo and behold, I had the opportunity to go to a clinical study with some of my colleagues, and we got there, and within the first 10 minutes, there was one of the site staff was writing on a tube with a Sharpie pen, and I actually thought Scott had like called ahead and set it up as like a
Right.
demonstration, but no, I realized like this is a real thing and this is really common. So
Yeah, and before we go on, let's pause for a minute and make sure the audience understands something, which is we are in no way, shape, or form undermining the important work that clinical trial researchers do. It's incredibly important work. These are often people who are very much overworked. You could argue maybe even underpaid. And we, as sponsors, I put myself in that bucket, we give them very complicated protocols and very complicated lab manuals to work through. I'm only saying that the current state of the art is not where it needs to be. And we need to be able to offer tools to help those clinical trial researchers to do their job more efficiently and even better.
Yeah, that's a great clarification, Rick. And it also seems like protocols are only getting more complicated as time goes on. So this problem is likely not going to reverse on its own. I'm curious. You mentioned a moment ago talking about essentially the sample switching that happened in your previous experiences. Could you talk a little bit more about kind of the downstream effects? Like what does that kind of mean for potential outcomes in these trials?
Yeah, for sure. And Brooke, thanks for your comment, and so glad you joined. But Brooke, for others that don't see it, noted that it's really the process issue. It's not the sites or not the people. These guys are showing up every day trying to do their best for patients. So thank you.
Yeah, well said. Absolutely.
Yeah, so here's the thing, Tim. Let me give two examples actually. I'm gonna start with one, and I'm sorry, I'm maybe gonna go into nerdy scientist mode for a minute. But this is my training as a pharmacokineticist. So think about a trial called a bioequivalence trial. All right. Now, so we've already talked a little bit about how these sample issues that can happen at sites are really important because in addition to causing quality concerns with regulators during review, they can actually mean the difference between winning and losing on a trial outcome. Like really, it can make the difference between is it positive, is it statistically significant, or is it negative, which is a, pretty big deal. So I mentioned bioequivalence study. So what is that? Let's think about a study where a sponsor is introducing a new formulation. So you've got a drug that's already on the market. They're going to change the formulation of that drug, probably for life cycle management reasons. The reference drug or the original drug is going to come off patent. If you introduce a new formulation, then there's a possibility to extend the patent. All right. These bioequivalent studies have a very strict statistical test or criterion that needs to be met. So the way you run the study is you dose healthy volunteers or patients with the test and the reference product. So the old formulation, the new formulation. You look at their pharmacokinetics. So biological samples are really important here in this trial design. And the statistical criteria are such that the geometric mean ratio of those two results has to be in a reference range for the 90% confidence interval of 80 to 125%. So they can't, again, it's called bioequivalence, not bio close. It's bioequivalence, right? And so if you're not within the confidence interval of 80 to 125%, that's a failed study. If you are, the study is positive, and that means that new formulation is equivalent to the prior. So imagine this, and you know, in my career, which has been pretty long and extensive, plus all kinds of market research we've done as part of TruTechnologies, plus being in business for 6 years, I'm
Yeah.
confident in saying that around 20-25% of the time, there are issues with biological samples in clinical trials that they're lost, switched, mishandled, mislabeled, otherwise unusable. 20 to 25% of all those samples have issues. Now, think about what I just said about the strict statistical criteria for BE. What if straight off the top, you're throwing out 20% of your data, adding
Yeah.
20% variability when your window for equivalence is this? Like
Yeah.
you are significantly putting your trial at risk where biological samples are the most important thing in that trial. Okay. I mean, we were talking, so the survey earlier talked about development timelines and cost. Think about this. Do you really want to blow a BE study such that you're having all this sunk cost in that new formulation development and then either having to go redo it or just say that I just ate like $12 million dollars?
Yeah. Interesting. That yeah, that is powerful, Rick. I want to, if we can go back, you had mentioned a couple of moments ago kind of the ethical concerns. I wonder if we could talk just a little bit more about that. I know, we're talking about, and I think sometimes it's easily said as we're still talking about human beings and we're talking about, actually collecting samples from human patients and in some cases healthy volunteers. I know that this is a personal priority for you and that the ethical side of this is something that's top of mind for you. Could you talk just a little bit more about that?
I can. I I gave the bioequivalence example. I did also want to just briefly mention before we move on to the ethics,
Oh,
another
yeah, please.
another example. I won't go into detail on this one, but I just want to make sure the audience gets to hear this because I think it's really important. A lot of times we run clinical trials with some objective efficacy measure, maybe tumor size measurement or RECIST criteria or something else like that, imaging. And but other times we actually run clinical trials, even phase 3 trials, where there's a biomarker as the primary endpoint of the study.
Yep.
So think about what I said for the bioequivalence study, which is a special niche situation, but think about where we're running a phase 3 trial with a biomarker as a primary endpoint. Do you really want to take a chance that your trial is going to fail because of samples being switched? I've done some work, Tim. In fact, I kind of wish we could share slides on this, but maybe for another time. Um, but I've done some work with a biostatistician just to look at the impact of sample switching in a trial where the primary endpoint is a biomarker. And it was actually unbelievable to the statistician when he did it. It's sort of unbelievable to me, but it all makes sense. If you assume 20% sample switching across arms in a randomized trial where there's a biomarker as a primary endpoint, you're going to take your phase 3 statistical power from 90% down to as low as 40%, depending on the degree of sample switching and how much is across arms or within arms. Nobody in their right mind is going to run a phase 3 trial powered at 40%. That means if the drug is positive, if the study is positive truly, you have a 60% chance of being wrong.
Yeah.
Okay. So anyway, I just want to get that out there.
No, I
The
appreciate that. And on that topic, while we're still talking about it, Rick, I'm guessing that if these type of operational issues or even like actual sample switching, like when would you even be aware of this from the sponsor's point of view? Like how far down the road before these issues would even come to light?
I mean, that's the crux of the problem, right? Because not only are there issues in the way that we capture these important data at the
Right.
sites, there are also issues in the lack of visibility to the sponsor. You sometimes you know at the end of the study, unless you're doing an interim analysis, which isn't all that common, especially in phase 3, then you're going to know at the end of the study,
Yeah.
or you're never going to know
But I was
because,
going to say there's also a
or
world
you're
which you
never
really
going to know.
don't ever know. Yeah. Yeah.
Okay. Anyway, I'm
Awesome. Can I pull
sorry.
you back into
Yeah.
the ethical question if you don't
Yeah,
mind?
thanks for letting me go back to
Yeah,
that.
of
Okay,
course.
yeah, I mean this is a pretty big deal, right? Like I'm talking about the practical aspects here on trial outcomes, which is also important, but let's just think about this for a minute. Um, the ethics that we're talking about here with regard to biological samples are this is just a result of outdated processes. It speaks for itself. Like that's sort of why does it happen? Well, it's outdated processes.
Right.
In my opinion, it's unacceptable. I mean, think about this, Tim. Imagine one of your friends, family members decided to participate in a clinical trial, and they're doing that for probably two reasons. One, to be altruistic and advance clinical science and medical
Yep.
research. The other is that if it's somebody who has a condition or a disease that they have a chance to benefit from, they're going to take that chance and do it.
It's hope, right? It represents hope.
It represents hope, but they are taking on significant risk
Yeah.
still. That, you know, in a clinical trial, that's usually a therapeutic that hasn't been approved.
Right.
Um, what if they were told that because a site is not using available technology to actually minimize these issues, that there's a high likelihood that one out of every four blood samples or tissue sample that's taken from them is basically going to go in the garbage? Like, is that acceptable? I don't think so.
Yeah. Yeah.
You know, I get I'm going to try not to show it, but I do get relatively fired up about this. I mean, these oftentimes are sick people, again, taking time away from sometimes the limited time they have left, and we owe them more, and we can do better
Yeah.
than we're currently doing.
I think it's an issue worth getting at least a little bit fired up about. Yeah, that, no, I appreciate that. And thanks for
Yeah,
the thoughts on that.
yeah.
Why don't we?
So hey, just to wrap up on that, I
Yeah.
mean, just to make it really clear, um, I this is why I'm dedicating my time, a lot of my time, to make this a priority right now. Um, it's great that others are focusing on AI and drug discovery. I don't want that to stop. I want them to keep advancing. It's great that people are focusing on using really sophisticated tools for data analysis. I want that to keep going too. It
Yeah,
helps
of
all of
course.
us. But
Yeah.
I'm going to be the advocate for fixing a really simple problem and one for which we've already created a solution.
Yep. No, I appreciate that. Let's jump into another poll question. This is going to be our second poll here. We have three possible choices, and the question is, how confident are you that your clinical trial sites execute your protocols as intended and record data accurately? And our choices are very confident, somewhat confident, or not at all confident. And yes, this is a mildly provocative question, and we appreciate your participation. We're going to let folks have a chance to respond. We'll give it just a moment here, and then we'll take a look.
Tim, you're a fantastic moderator. Thanks for making this easy.
Oh, thanks, Ray. That's my pleasure. We're not done yet. Well, let's not celebrate
Okay.
before we get to the
Okay.
finish line.
You're going to throw me some zingers?
I got a couple curveballs. Let's see how we did here. Let's grab some results. Okay. Probably not a shock here, Rick, but the vast majority of our respondents are saying they're somewhere in the middle, right? Somewhat confident. I'm going to throw it to your thoughts. I would say it does seem noteworthy to me that we are looking at least 30% of folks saying, or just under 30% of folks saying that they are not at all confident. So I guess if you look at the top bucket, Rick, there's only 7% of our respondents saying that they're very confident. Are your
Yeah.
thoughts on that?
Yeah, they're right. I mean, I'm glad we've got an audience that gets it and they understand. So let's do better.
Yep. Yeah, definitely in line with the conversation today here. Well, excellent.
Hey Tim, let me just respond a little bit more thoroughly to that if I can.
Sure.
So
Yeah.
so talking about how confident are we that sites are executing the protocols. Think about this for a minute. You guys on the webinar know this. You're all, I assume, all at biopharma or pharma companies for the most part. Think about how we do this. We as a group, and I'm putting myself in that bucket. I'm not dissociating myself from you guys. I'm a drug
Right.
developer as well. What we do is we write really complicated clinical trial protocols, and they only get more complicated as advances happen in science and medicine. We write them kind of by committee. We have a bunch of authors, and we do our best, but it's always fast paced. It's always in a hurry because we're trying to move quick. And then we'll send that protocol to one of many sites around the world. So let's say there's 50 sites in a phase 2 or phase 3 trial. Then we've got another group of people, a little bit of a Venn diagram with authorship, but sometimes different authors writing a lab manual,
Yeah.
which is also very complicated. These are documents that, if you're lucky, like a really good one might be 100 pages,
Right.
but they're probably 130, 140-page documents. And then we're sending lab manual to all these sites. And guess what we do? We basically ask them to 50 sites, reconcile those, interpret them, and understand what happens on a given visit on a given day for a given patient.
Yeah, and I know from our team's experience here, it is not uncommon for there to actually be some discrepancies between the protocol and the lab manual, right? So that adds, in terms of you mentioned that reconciliation, that adds another step, right? Like which one am I following here? So yeah, incredible. Rick, I want to kind of keep going a little bit forward looking and maybe a little bit backward looking, but when you and Scott, started out building True Lab and now TruTech, relative to sponsors, like what were you hoping this would change for how sponsors oversee, particularly from an oversight perspective, sample collection and handling?
Yeah. Yeah. I mean, if you kind of think about it, it's really like, remember when I said Scott didn't believe me? What it is it's moving away from how things have been done probably since the 1970s or earlier and using contemporaneous methods to do better. That's really the bottom line.
Yeah.
Um, but let me just orient the audience just super briefly to explain what TrueLab is, the main product of TruTechnologies. We've kind of talked around it, but I think it's important to just clarify that. This is
Yep.
a mobile technology built for sites, sold to sponsors, but built for sites to make their job easier. And it captures biological sample information as source data. So it's interesting in fact, I was telling you guys, Tim, before we started the webinar that I was on a call yesterday for a panel that I'm going to be on at a future conference. And I briefly explained this to people who had never heard of it. And they're like, well, wait a minute. What about the EDC? How do you do source data verification? And I'm like, wait a minute. You guys got to like think differently. You're always thinking about what you're used to, and that's what
Right.
we do, right? We think about what
Right.
we know. This is different. This is a very simple mobile technology that captures source data. It's on a device that scans a barcode that's on a tube and captures pseudonymized patient ID, date of collection, date of dose, time of dose. That's pretty much it. In addition to the fact, remember we talked about these disparate protocols and lab manuals. We
Yeah.
have a digital workflow for sites to make their job easier.
Yep.
Okay. So that's kind of it. Now, so that's what it is. Now you asked something different, which is what did we try to change? So let me tell you something here. This is kind of a nice at least an analogy for me to help understand it. I remember when I was an undergraduate, I took an animal behavior class. Pretty typical for me. I wasn't sure what I wanted to do when I grew up. I was going to be a vet at one point, and I learned about this classical animal behavior study. So these birds, birds usually fly south for the winter, right?
Yep.
So there's this animal behavior experiment that was done where these birds were magnetized, so their polarization changed from north to south in their brains. Okay, at a young age, and that made them fly north instead of south for the winter. So pretty cool experiment, right? And I'm pretty sure that when I was a baby, I was magnetized because my sense of direction is horrible. And if I'm going to go somewhere, I usually just go the opposite way, and I'm pretty close to where I need to be. All right. Then um, I remember when Garmin introduced the Garmin Nuvi, this
Yeah.
portable device. Now all of us have these like in our cars for the most part now, but this portable Garmin, my gosh, that was a game changer for me. No more paper maps, no more drawing out a route when you're going on vacation with your family, no more being late for appointments, driving down the highway and oh my God, I'm the wrong direction, right? So here's my analogy. My analogy is that clinical trial sites are using paper maps to run their studies. TrueLab is the Garmin Nuvi for sites and sponsors.
Oh, I love that. Yep. No, that makes a lot of sense. In terms of monitoring risks around these samples, like how does capturing this information digitally right at the source, how does that change the way that sponsors can monitor risk going forward?
Yeah, I mean, it's super important for people to understand. What I didn't know when I first started thinking about this years ago is that the majority of the mistakes with biological samples happen right there at the point of collection at the site.
Yep.
It's not,
Yep.
I used to think it was a bioanalytical lab. Like when I would get data, I feel so bad for these guys now. I should probably write them all an apology letter. But I would get data and I would call the BA lab and say, hey, you guys must have made a mistake on your instrument. Can you rerun these samples? Clearly, the peak is where the trough should be, and it's not right. And they'd
Yep.
rerun them, and I would get the data back to like the fifth decimal place exactly the same. So what I learned is that I would actually bet that it's 90% of the mistakes that happen are happening at the site. So capturing data as the source with a very simple device that is easy for sites to use is a game changer. There's no more asking these technicians or research coordinators or nurses to look at a clock, write down the time on a piece of paper, later enter it into an EDC where mistakes can happen. So this is now the single source of truth. And it doesn't just prevent the errors outright, it also remember we talked about time and
Right.
cost of trials?
Yeah.
It gets you to top line results faster because you don't have to deal with all this query resolution. That's become status quo in the norm in clinical development.
Yep. So, in terms of the sites themselves, the sites have had, I don't think it's any secret site, have had a lot of tech pushed on them over the years. I think that there may be even some natural resistance to new stuff, right? Oh, this is the latest and greatest thing that you're going to use. So I know one of the kind of the mantras that we talk about here at TruTech is that you really got to bring some value to the site experience, where you got to deliver something to them to make their lives easier if you're really going to get them to adopt. So can you talk a little bit about some of the ways that this can actually make the site experience, not just for the sponsor's perspective, but also for the actual site users?
Yeah, absolutely. And just to kind of reiterate a couple of things, I have huge respect for the people that are doing this work at clinical trial sites. I'm
Yeah,
friends with many of them. And so,
absolutely.
I spend time with them. I know how hard they work. I know what they're driven by. And it's really to help these patients, right? So huge respect for them, which is why before we embarked on building anything, we started out with the idea that we have to make this something that is easy for sites to use and make their job easier. You mentioned technology burden. It's true. And again, I'm not going to like point fingers. I've been part of this. I've run trials in the past where we had technology that was not patient centric. And it wasn't, in hindsight, it's probably not something we should have done. And I don't blame sites for resisting.
Yeah.
Tim, this is an app on a phone. I mean, this is
Yeah.
not technology burden. Okay.
Yep. Yep.
So how do we do it? I talked about source data capture. Okay, so not only are we making sure that we're capturing really important data that's correct at the outset, guess what? What's new here is that sponsors can have a dashboard and instantly see what's happening at the site instead of waiting till the end of the study or weeks later or trying to call the site constantly and get a spreadsheet or, an email. Okay. The other thing I briefly mentioned is this digital interactive protocol guide. This is not a PDF scan of a 200-page document. This is literally just walking through step by step.
Or the handwritten cheat sheet is another common one, right? Like
Yeah,
the
but now
greatest
we
hits
but
of
now
the,
we've
yeah,
replaced that. This is point your phone, scan the barcode, tap, tap, tap. There's very little data entry involved.
yeah,
There's this automated workflow process where you can integrate the protocol and key tasks into this one simple platform. And then, we don't have time to go into a lot of detail here, but I'm happy to talk with anybody separately on this. On our back end, meaning not the site-facing app, but on our back end, we receive a tremendous amount of data. I mean, the app is used in almost every country around the world in the local language. I can't even tell you how many sites we've been deployed to now, but we have a tremendous amount of data, which means we, TruTechnologies, has an obligation to make this simple for sponsors as well. So we have a risk-based monitoring engine that we employ, and we have escalation mechanisms to the sponsor that if something's happening at a site, and even down to the level of the user, Tim, at a site, we can tell you what might be going wrong at a particular time. And you can intervene in real time. This prevents protocol deviations, and in some cases, it's actually saved a study from going sideways.
yeah. It is also a completely new way of thinking about this, right? Particularly from the sponsor perspective, like being armed with this information. I think we've had some sponsors be like, "This is great, but like I've never really had this before." We talk sometimes, Rick, here about turning the light on the room, right? And sometimes
Yep.
there's some surprises there. And I think sometimes the challenges are like, well, how do we action this information, right? We've never had this kind of visibility before.
Yeah, and I would much rather know when it's in my control than when somebody else is looking at it and determining whether the drug should be approved or not based on how I conducted the study.
Yeah. Absolutely. Well, Rick, this has been a pleasure as always. I think maybe to wrap up, if you don't mind, could you maybe pull it together for the audience to just kind of summarize into some key takeaways before we wrap up the session today?
Yeah, again, thanks for moderating. Thanks to everybody for joining. If I had to bucket it maybe into three key takeaways, one is, as you said in the outset nicely, trial success is not just a function of the biology and pharmacology. That's really important, but it's also a function of how we conduct the study. Conducting the trials with surgical precision sometimes can mean the difference between a positive and negative trial.
Yeah,
So let's use the technology that's available and that sites enjoy using to make their job easier, to increase speed of these trials and increase the probability of success.
yeah,
Um, I'd say good enough is not economically viable with all the intense competition that we're talking about. So you were just talking about this is something new and different. Yeah, change is hard, right?
yeah,
There's a classic book, Who Moved My Cheese, right? We contemplate, we hem and haw over these things. Well, let's just stop and think for a minute and realize how things are being done and that there's a better way to do them.
yeah,
And then finally, let's be respectful of patients. Like that's really a big ask that I have here. Again, like I've chosen at this point in my career to dedicate my time to do this. I'm an optimizer, right? I want to make it better for sponsors. I want to make it better for sites, but it's really, really important that we're doing the right thing by patients. We owe it to the patients that are participating in these trials to move away from how things have been done for so many years. If I'm not wrong about this, I think the first randomized clinical trial was done in the 1940s. It'd be interesting to see, how different is it really? I don't know.
yes,
But
yeah,
but 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 let's use the technology and tools that are available now to fix things at the point of collection, make it easier for the sites to do their job, make the data better for the sponsor, increase speed, decrease costs, and increase probability of success.
yeah.
Okay.
Great. Terrific. Well, Rick, it is always a pleasure. Great to talk to you. Thanks so much for spending some time with us today. I want to thank everybody for joining us. Thanks for your attendance and your participation. We really appreciate it. Just to let everyone know, there is going to be a recording of this that will be available. We'll send that out to participants shortly. Otherwise, thank you so much for your time. We appreciate it. We hope to see you again on a future session, and we hope you have a great rest of your day and rest of the week.
Yeah, thanks everybody. Thank you, Tim.
Take care. Thanks.